Hello everyone, just wanted to clarify something about the machine learning algorithms that we have on the platform. A quick intro before I jump into it. In the platform, on the T-BioInfo platform, we have these data mining sections called supervised analysis and unsupervised analysis. So again, what's the difference? Unsupervised analysis would be things like clustering, PCA, where you would kind of explore the data and identify data structures that you want to analyze. Under supervised analysis, we have classifiers and different methods that would help you work with the data that you can label. Okay, so the biggest difference is that if you don't know what your samples are, you would want to use unsupervised analysis to explore. If you know some specific things about your samples, for example, if you know a class, a subtype, or something like that, then you can use the supervised analysis section. Okay, so I know that a lot of you had questions about how to exactly use a supervised analysis, and we recently updated this section. So I just want to show you how to prepare the files and run the analysis to get results. And what kinds of results would you actually get? Okay, so I have here a couple of files. One I call test, and another one I call train. Now in the new updated version, you actually don't need the test and train, but let's keep it because it will still be easier for us to use. What's the difference? Well, the difference is that I have the sample name here on top, ID, so this is a table of expression, and this would be something that you get from the RNA-seq section. So in RNA-seq section, we process the data and we got a table of expression. We have numbers in here. Remember that normalization, log scale, and what exactly are these numbers is going to play a big role in your analysis. But we got some table of expression. And what are these? These are gene IDs. So these are going to be our features. Again, what's important here is the number of features. The more features you have, the more samples you should have to really work with these features. Because you might run into overfitting. But right now I'm just going to try and do something that might not make a lot of sense, but at least technically will work. So now here I have a row that's called class, and the class is really my labeling of the samples. So this is different types of cancers that I have labeled here. I have a bunch of different samples. Okay, remember class ID, not group, not name, not ID, not anything like that, class with a lower C. Alright, so let's save this. What is in my test? Okay, so theoretically, what is a classifier used for? The classifier is used to classify some set of samples based on a known set of samples. So I have, let's say, 5 known samples and then 3 unknown samples. So I can train the model using known samples that are annotated or labeled, and then use that model to classify new samples. But a lot of times what we see in these kinds of experiments is the need for feature selection. So I want to select the genes that are responsible for separation between different classes. For example, I have some classes that are cancer, some classes that are normal. Could I use a classifier to select those features? Yes. Some versions of classifiers are what's called greedy algorithms. They also select informative features. And others, for example, like random forest, which are decision trees, they're not really classifiers, but they could be used as classifiers, they also give us some evaluation of features inside. So which features were used to make those decisions at different trees. Okay, so let's try and run this and see what we get. Okay, so on the platform, go to areas of analysis and select supervised analysis. Now let's upload both of these files. So here's the test, here's the train, upload it, wait until it's fully uploaded, define groups check, okay? This is the training group, this is the test. Okay, what's the difference? Just one again. Training is going to contain the labels for samples, and the test is not going to contain it. And of course it will vary, it will be up to you what exactly you put inside. But remember, your gene names, if those are your features, they have to match exactly. The number of features plays a role. The numbers in the cells also play a role, because some of these methods are actually going to be assuming different things about those numbers, and if your data set does not fit those assumptions, it will fail. Okay, but for now, let's click on Continue, Start, and let's select Stepwise Lda. So Stepwise Lda over here is a stepwise linear discriminant analysis, is used to find a subset of the provided features that optimally separates the classes inherent in the data. Okay, so this is a description that you can read more about, but let's look at this picture right here. So this is somewhat like PCA. You have two axes, LD1, LD2, and LD3. So you have different combinations, LD1 and LD2, LD1 and LD3, LD2 and LD3, etc. And within, you will see your sample names by class, and it will show you how it separates those different classes. Okay? Now you have here this parameter nouveau. What does it actually do? Well, it's an internal parameter that is used to draw that line that separates the samples. Typically, you would just want to add another 0 here if you're using the type of data that I just used. And now you can click on End, and then you will save your pipeline. Save. Okay. It's called Lda Stepwise Lda. Test. Save it. Run it. Okay. I already ran this pipeline, so let me just show you what you get. Stepwise Lda. Let's look at pipeline output files. What do we have here? So we have a number of files. The first one is a plot. Let's take a look at what is in the plot and discuss a little bit what we see in here. Okay, so these are all of these different cancers that we were looking at. Now I didn't really normalize the table or do anything with the table. I just ran it the way I was given it just to test this. But you can see that the separation is actually not that great. However, if you look further into this pipeline, let's see what else do we get. So for example, coefficients. These are the coefficients for features. So let's open that file. And let's see what are these features. So these features are going to be genes, and let's see how many. We have 50 features. Okay, so this is a lot, but nevertheless, we can now take this list and we can go back to our expression table and actually find them. Okay, so let's take a look. For example, the first one. This is, let's find the training data set and just search for that. So here we go. I found this sample, and I'm just going to mark it right here as 1. All right, why? Because it's going to be easier for me to find it. So let's insert really quickly. Okay, and let's see, why did it choose this as a good gene? So let's select data, and let's just label this data with the row names, which is going to be class. So let's go to the very top, and this is going to be our label. Okay, and press OK. All right, now let's go back to the chart. Let's take a look. Okay, so we already saw that it's not a perfect separation. Nevertheless, we do see some interesting pattern here. So for example, colorectal cancer, which is all right here, is all upregulated. Breast cancer is all pretty much underexpressed. This cancer, AMP cancer, is also somewhat higher. Pancreatic cancer is higher. So you do see some insight into the cancers that you're studying using this gene. Now, how would we choose this gene? Not in the same way. We could do differential gene expression. So obviously, if we took colorectal cancer versus breast cancer, this would probably be easy to find using something like t-test or differential gene expression. But here we actually get more information. It's not just about a pair, it's about multiple types. And we have about 50 genes that are going to be informative for this kind of separation. Okay, so I hope that this was informative in terms of preparing your data and in terms of running something like Stepwise LDA. Of course, remember that selection of features and what kinds of numbers you have inside is all going to play a big role in how successful you are at getting results. Now, if you have any questions, make sure to check out the chat box. Type us a question, reach out to us by email, and make sure you take the courses. Transcriptomics 3 talks about this procedure a little bit in greater detail.